everyone and welcome to my channel Be Yourself. This is Dr. Rajni Sharma and today I'm discussing the second part of the part C from the June 2015 questions of CSIR JRF Net Life Sciences. In the last video I have discussed questions up to 100 and now I will proceed after that. So the question number 101 is that carbohydrates synthesized by photosynthesis are converted into sucrose and transported via phloem to other parts of the plant. The following aspects are associated with sucrose uploading in phloem and its transport. Yes, we know that after the synthesis of sucrose, it is uploaded in the phloem and it is transported and it follows both symplastic and apoplastic pathway. Another is that the route which is followed by starch is that it will be first uploaded from the phloem towards the mesophyll cells, towards the phloem parenchyma, towards the companion cells, and finally it arrives to the sieve tubes. So this statement is also true, and obviously the transportations in the sieve tubes will be as per the pressure flow models because it always flow from the higher concentration toward the lower concentrations, and the pressure which is basically created by the transpiration pool which also pull up the solid particles towards the required place is also controlling the upward movement or the transportation of starch in case of plants. So here the answer will be B, C and D. So as per this, answer is number 2. Next question is symbiotic nitrogen fixation in legume nodules involves complex interaction between rhizobium and legume roots. This complex interaction is governed by actually the nodule formations in case of rhizobium is controlled by a node gene. There are basically three types of node gene, A, B and C in case of rhizobium, and also the host cells also has some node genes. It can be E, F and H. So the first statement, number C statement, activation of node gene in rhizobium is true. Another is that what happened, there is an integration of plasmid of rhizobium to the root nucleus genome. After the integration of the genome of rhizobium in the root nucleus genome, it also accelerates the synthesis of nitrogen also. So this statement is also true, and obviously the activation of nodular gene is in legume roots. And the factors required for the activation of nodular gene is present in the legume roots itself. It can't be activated outside the root. So the statement A, C and D is right. So the answer will be A, C, D, number 2. Next questions, quite easy questions, and this I found these questions repeating all the times. So and it's also easy to remember the cycles of pigment system 1, pigment system 2, which is also known as cyclic and non-cyclic phosphorylation. So don't miss this topic because this is very important. Every time you will find, and the same pattern of questions has been asked every time, and it is in 4 marks. Can you imagine? It's such easy questions in the 4 marks. So better don't relieve this topic. Mug up this cycle. It is a jet scheme. A jet scheme describes electron transport in the jet scheme is what? It's a non-cyclic phosphorylations. Transporting oxygen involving photosynthetic organisms. The direction of electron flow is presented in the following sequences. What happened? The pigment system 2 or pigment system 680 will pass its electron towards the pheophytin to QA, QB and then to cytochrome B6F and then to PC, and this PC will transfer its electron toward the pigment system 1 or pigment system 700. So this sequence is wrong. This right sequence is here, where the pigment system 2 is passing its electron to pheophytin, then to QA, QB, then cytochrome B6F, then finally to plastocyanin, and it finally transfer it to the pigment system 1, which is 700. And now from the pigment system 700, the electron will pass to the phyloquinone. It will then pass it to the ferredoxin Sx, Sa, Sp, and finally to ferredoxin. And in the case of if it will be cyclic, then from the ferredoxin it will be passed toward the cytochrome B6F complex, and then it will again recycle to pigment system 1. But here it is a Z scheme or the non-cyclic phosphorylation. So the first electron will be transferred from pigment system 2, and then finally it will reach to the pigment system 1, where it will get reduced with the help of NADPH. So the C and D is right statement, and the answer will be number 3. Next question is, following are certain statements related to plant exposed to dehydration stress. So what will happen to the plants in the dehydration state? Not only in plant, but in case of human also, what happened during the dehydrations, we have to take up more and more water. So plants will also will adapt in such a situation, or will adapt itself in such a way so that it can absorb more and more water in itself. So what will happen when the water potential of the rhizobium decreases? Sorry. Due to water deficit, plants can continue to absorb water as long as plant water potential is lower than that of soil water. It's true, it will be created with the help of transpiration pool, as in the dehydration state, transpiration is also one of the major cause for the dehydration state of plant. So, and the next is that the ratio of root to shoot growth increases in response to water deficit. Actually, what happened in case of water deficit, plant will adapt in such a way that its root can penetrate as long as towards the groundwater, so that it can absorb more and more water from itself. And another thing is that another modifications what plant can do, it can develop a hairy structure around the roots, where it can hold, or it will not miss any of the droplets of water from the soil. So these two adaptations generally plants suffering from dehydration, or the plants which are prone to the water deficient areas, adapt in such a way. Another point is that, so the answer A is true, B is true. Another point is here, plant cells tends to release solutes to the lower water potential. No, it's not possible, because always solute, if there will be lower water potential, then in the case of plant cell, if it will have the higher solute concentration, then it will not release solute, rather it will uptake water molecule from the surrounding to make it rehydrate. So the statement C is wrong, but the abscisic acid is synthesized at the higher rate when the leaves are dehydrated, and the more abscisic acid accumulates in the leaf epiblast. It's also true. So the statement A, B and D is right, and the answer will be number 3. Move to the next one. A cross was made between HFR, which is the high frequency recombinant, methionine plus, arginine plus, leucine plus, and streptomycin sensitive, when it is crossed with the F minus, which means it's a recipient bacteria having all the sequence substance, but it is resistant to streptomycin. In which conjugate leucine plus is X conjugates are selected. X conjugates means first of all there will be the transfer of leucine, or we can say leucine is the first gene which is transferred to the bacteria, to the F minus bacteria. If the linear organization of the gene is leucine plus arginine methionine, it means leucine followed by arginine and methionine. Which one of the following genotype is expected to occur in the lowest frequency? The lowest frequency will be number 4, because if leucine is followed by arginine, then it will be methionine. Then is it possible that leucine first, when conjugated or when it is transferred to another bacteria, but it will be always followed by arginine? But instead of here arginine, you can see that methionine is transferred to the F minus or the recipient bacteria, but arginine is not transferred into that one. So is it possible? It is not even possible. So the lowest frequency, if it somewhat happens also, so it will be in the lowest frequency. The answer will be number 4. Move to the next one. Two homologous individuals P1 and P2 were genotyped using dominant DNA markers A and B as shown below. The F1 progeny obtained was test crossed. The frequency of progeny with which different genotypes appear is given below. The one having the frequency for A gene is present only in P1, for the B it is present only in P2, which proves if it will be in the same sequence, then it will be in the series form. But as these are on the both opposite sides, so this is on the transform. And the progeny have 45 having the parental one, and we know that the lowest frequency is that of the recombinant one. So the lowest frequency, which is the recombinant one, has the frequency only 5. So this means if we will have to calculate the distance between the progeny, what we calculate the total number of progeny. So here the total number of progeny is 10. So as there are 5 progeny having the A marker and 5 progeny will have the B marker, so in combined it will have the 10 that's the distance between this recombinant will be 10 centimorgan. So move to the question. The answer will be in the F1 marker A and B are linked and is repulsive phase or in the trans phase, and the distance between A and B is 10 centimorgan. So the answer is B and C, which is number 3. Yes, next question is from the pedigree analysis. The above pedigree shows the inheritance of the rare alleles. The allele is is very rare, and it is here showing that rare allele is the female one, which is from the outside, not in the from any of the generation. So the allele is X-linked and autosomal recessive or dominant with the incomplete penetrance. As it is the rare allele, so it can be possible that it is incomplete penetrance. So here, if it is showing, if it will be dominant one, then completely dominant, then it will have been expressed in just F1 generation, but there is no offspring in the F1 generation is affected. So it is definitely it is not dominant. But it is also possible that if it will be dominant, but there will be incomplete penetrance. Incomplete penetrance means the one ratio, suppose we are expecting that if it will be dominant, then there are 5 individuals should be affected. But we are having only one individual get affected, which means that there is a penetrance of 5, but it has only one individual which is affected, which means it is not completely penetrating, rather it is an incomplete penetrance. In case of dominance also, it is possible if it is not expressing in just F1 generations and it skip the generation, it may be possible that either it can be recessive one or it can be dominant, but it will be have incomplete penetrance. Here it will be incomplete penetrance with the dominant one because it's a quite rare alleles. So the answer will be dominant with incomplete penetrance. Next question is, the plant with the red fruits is crossed to a plant with the white fruit. The F1 progeny had red fruit. On selfing the F1, 2 kinds of progeny were observed: plants with the red fruits and those with the white fruits, to test whether it was a case of recessive epistatic interactions. A chi-square test was performed. The value of chi-square is 1.062 was obtained at the p-value of 0.05 or at the value of 5% significance. It is 3.841 given. So here the value of chi-square is less than that of the 3.841. So the hypothesis which we will take, it will be accepted in this case. So let's see what should we take the hypothesis, and the whatever hypothesis we will take, it will be accepted because the value is lesser than the significant value at the p will be significant one. So what the hypothesis we will take, as it's the case of the recessive epistasis, so we will take the null hypothesis as the plant was with the red and white fruit ratio will have 9:7, and we will accept it as the ratio, as the value of chi-square is less than that of the 3.841. So the answer will be based on the chi-square value. It is case of recessive epistasis interaction. Yes, it is a recessive epistasis interaction. So the answer will be A and C, or number 1. Yes, next question is, the somatic recombination was caused by mild exposure to radiations on flies heterozygous for a given allele during the specific stages of development, and the individual allowed to develop. Such individuals are likely to have, if it is, first point is that it is a somatic recombination, and other is that it is, and the flies are heterozygous. So it is possible due to recombination that heterozygous may convert it into the homozygous state. So as it's a somatic recombination, so there we can have the patches of mutations. Not all the genes will be mutated. So there may be possible that twin spots, or the patches of mutant cells, and the homozygous wild types cells in the heterozygous body will be there, and the other is the first one, which is a clone of homozygous cell in the heterozygous body. Both almost are same statement but in different way. So the answer will be A and C, or number 3. Next question is, the question is quite simple, but it is looking quite complicated. The question is, sickle cell anemia is a recessive genetic disease caused due to the point mutations in the 6th codon abolishing one of the MSP2 endonuclease digestion site present in the beta globulin gene. MSP2 digestion DNA from the normal person give two bands. One band will be 1150 base pair, and other band will be 200 base pair. In beta globulin gene, a family will be proband. So based on the disease phenotype, gave the following MSP2 digestion pattern. The one in the normal case, what we will have two pens, one in the 1150 base pair, and other will have the 200 base pair. So likewise here, in case of mother, we have the 1150 base pair band, and other is the 200 base pair band. So it is the normal case. And in the father, having the one gene for the normal, and other having additional gene, so it can be possible it will be the band of the abnormal myoglobin. So we can say that it's a carrier. But in son, the normal one is completely absent, so it can be carrier. Son is affected one. Daughter is also having the same pattern with that of the father, so it is also the carrier one. And the son number 2 is normal one. So move to the question, options. Son 1 in the proband, and the given mutation is not present in the son 2. It's true. Other is daughter is the carrier for the given mutation. It's also true. The gene is X-linked. It is not X-linked, because if it would be X-linked, then it will definitely present in, daughter will be the affected one. So it's not the sex-linked. Other is the father, mother, and daughter are affected. They are not affected, rather they are the carrier. So as statement is strong, the de novo mutation in the same site on the normal gene has allowed the appearance. It may be possible that some of the mutations has appeared, so that normal alleles may have appeared as a diseased phenotype. So the statement will be A, B, and E, and as per this, the answer is number 1. Number 111. In high altitude, hypoxia induces increased number of circulating RBCs, which can be explained by the following changes. During hypoxia, what happens? There is the increase in the production of HIFs as well as erythropoietin secretions, and also there will be the increase in the myoglobin as well as cytochrome oxidase. But here the statement, as the question is asking, the statement which is wrong. So here the number C and D statement is wrong, which says the myoglobin and cytochrome oxidase is decreasing. So the answer will be C and D, which is the wrong statement. Instead of decreasing, it should increase during the hypoxia conditions. The mechanism of sound locations in the horizontal plane by human auditory systems can be explained by difference in time between the arrival of a stimulus in 2 years. Yes, the distance, as the distance of 2 years will be always different. So the time difference will also, though it's of very milliseconds, but it will be there is a difference in the arrival of this auditory sound in between the two years. And also there will be the difference in the stimulation signals, also timing of stimulation signals. Next point is the difference in phase of the sound waves in 2 years. Yes, there is a difference of, definitely, if the phase of timing is different. Suppose one after 10 milliseconds we have the depth, then in the uh, even if we will lag for a few milliseconds, so there will be a possibility that this wave have traveled to another half of the wave. So there will be the phase difference between the 2 years. And the other is that the difference in tuning curves of the two auditory nerves. No, there will be no difference in the tuning curves, rather it will be same. And the other is that the activity of neurons is superior olivary nucleus, but not in the neural activity of auditory cortex. So the statement right is here A and B, but here we are asking the wrong statement, so the answer will be number 4. Next question is, during physical exercise, oxygen supply to the active muscles is increased, which has been explained by the following statements. What happened where the partial pressure of oxygen is low, then hemoglobin will release hemoglobin will release oxygen toward the low pressure area. So the one where we have to deliver the oxygen, suppose this cell A need oxygen, so here the partial pressure for the oxygen should be less. So what will happen when hemoglobin will come in contact with the cells, because the low partial pressure of oxygen, this oxygen will be delivered to the cell. So the partial pressure of oxygen declines and partial pressure of carbon dioxide rises in the active muscles, so that there will be exchange of gases. Carbon dioxide will be taken up by the hemoglobin and oxygen will be released to the active cells. Next point is that temperature is increased and pH is decreased in active muscles. Obviously, if carbon dioxide is releasing, then the bicarbonate ratio will be decreased in the cells. So there will be the decrease in pH at the active muscles. Another is that 2,4-biphosphoglycerate is decreased in RBCs. No, it will be increased in RBCs, because of which there will be reduced in the activity of RBCs for the oxygen. Another is metabolites accumulate in the active muscles increases the affinity of hemoglobin to oxygens. No, rather it will decrease the affinity of oxygen. So here we are asking for the wrong statement. So the wrong statement is number C and D. Next question is, the fractional clearance of neural and cationic dextran molecules of various sizes through kidneys of rat is shown in figure below. Which of the following statement is correct? Dotted line is shown for the cationic dextran and solid line is for the neutral dextran. What happened? The kidney has negatively charged wall, so there will be the better clearance of the cationic dextran in comparison to the neutral one. So with the increase in the diameter, there will be less interaction with the dextran and the wall. So there will be decrease in the clearance of the cationic dextran. So the figure number 2 sorry, justifies the statement. The figure number 2 justifies the statement. So the answer will be number 2. Next question is the estradiol synthesis followed by two-cell 2-gonadotropin theory, where partial synthesis occurs in the granulosa cells and rest in the theca interna cells of the graphene follicle. Which one of the following correctly represents estradiol synthesis and secretion? What happened? The two cells to gonadotropin theory is justified the statement 1, and it is the pathway for the synthesis of estradiol from the cholesterol. So no need to explanation in this question. You just need to know the cycle, what it actually carry out. Next question is, which one of the following is the most match for the protected area of India? A biosphere reserve is Nanda Devi Biosphere Reserve, and national park is Rajaji National Park. Ramsar site is number 2, which is the Loktak, and the wildlife sanctuary is definitely it's the Chambal. It's not the Sundarbans. It's the additional one. Wildlife sanctuary is the Chambal. So the as per this, A goes with the number 3 and B with number 4, and the answer will be, yes, the answer will be number 1. Next question, the following table shows selected characters used in analyzing the phylogenetic relationship of the 4 plant taxa. The one which don't have any of the characters shows the most primitive ones, and as per this, the most primitive one is the fern. Number second is the T4 will be followed by T1, where it has only one xylem and phloem. It is the oaks, and the third is T3. It is the pines, and the most developed one is number 2, which is the hornwood. So the answer will be number 1. Yes, question number 118. It is the most interesting part of questions in part C. The question is that given below are the main characteristics of the few mammalian orders, match the number of the animals with the characteristic of their order. The 4 animals which is given here is tapir, lemur, earthworm, and pig. This is tapir, lemur, earthworm, and pig. Move to the question. The one which have the hooves with the even number of toes on each foot, and they are omnivorous. If you look at the figure, it has the even number of toes in each leg. This is pig and this is omnivorous. So A go with number 4, which is pig. Next is teeth consist of many thin tubes cemented together at ants and termites. This is somewhat the teeth are cemented together, and this artwork at termites and ant. So number B goes with artwork, which is number 3. Opposable thumbs, forward-facing eyes, well-developed cerebral cortex, and they are omnivorous. It is lemur. Its finger is like quite opposing, like that of the human being, and its front-facing eyes, and also well-developed cerebral cortex. Last is hooves with an odd number of toes on each foot, and they are herbivorous. And this is tapir, which has an odd number of toes in its leg, and it is only herbivorous. So the answer will be number 3, which is this one. Next question, which of the following options match the plant tissues type with its correct function is the vascular plants? We know that trichites are the chief water conducting elements in gymnosperm, whereas the vessel elements conduct water in case of angiosperm. Likewise, septube elements conduct food in case of angiosperm, and safe cells conduct food in case of gymnosperm. So as per this, it goes with 1, 2, it's for 2, and it's for 4, and this is for 3. So as per this, answer will be number 1. Next question is, which of the following gives the correct human diseases causing microbes match of the following? So it's quite simple questions. Even if you know the only 1 or 2 of the options, like chronic gastroenteritis, then we know that it is the causative agent is Helicobacter pylori. The Lyme disease is caused by Borrelia burgdorferi, and Lyme disease scarlet fever is because of the Streptococcus pyogenes, and typhus is due to rickettsia. So the answer will be number 1. Quite simple question. We have to match the following. And the answer is number 4, where it is Crustacea is the class, Hominidae is family, C or the Dermatoptera is the order, Ctenophora is number D is phylum, and Archaea finally it's the domain. So the answer is number 4. This question was quite simple. Yes, compared to K selection, R selection favors. K stands for king and R stands for rat. So obviously size of rat is small, and as for the R selections favors rapid development with a smaller body size, and it undergo reproduction more than once in their life. So such type of reproduction is known as heteroparous, while the one which undergoes only one time reproduction is known as simultaneous. So the answer will be, as compared to K, R selection favors rapid development, small body size, and early development with the heteroparous reproduction. So the answer will be number 2. Next question is, with which of the following is true about the digestive efficiency and ecological efficiency? Obviously, the endotherms have the high digestive efficiency, and ectotherms has the high ecological efficiency. Here the question is given the two legs 1 and 2 with the similar trophic structure of phytoplanktons, zooplanktons, plankivorous fish food chain were chosen to understand to top to down effects. Some piscivorous fish, those that fed on plankivorous fish, were introduced into Lac 1, making it a system with the 4 trophic level. 4 trophic level means something like this: 1 trophic level, 2, 3, and 4 trophic level. Where I was in the 4 trophic level. The Lac 2 has increased by adding additional quantities of nitrates and phosphates to study the bottom to up effect. One is from the top to bottom, and Lac 2 is to study from the bottom to top effect over a period of time. Changes in the biomass of each trophic level is measured. To increase the trophic level in case of one, what we are choosing here, some piscivorous insects are introduced, and this fed on plankivorous. So once this will be more, piscivorous will be more, then what will happen? In Lac 1, zooplankton biomass increases, because if this piscivorous is feeding on plankivorous, so the one which is dependent on zooplanktons will be decreases, or we can say if the amount of this trophic level will decrease, those who feed on this trophic level, then obviously the amount of this one will increase with the decrease in the number of its prey. So the biomass or the zooplankton biomass will increase, while the phytoplankton biomass will be decreased, because if there will be increase in the zooplanktons which feed on the phytoplanktons, there will be decrease in the biomass of phytoplanktons. So the answer will be number 1. Yes, quite easy questions and designed so beautifully. Here the question is, given below is the matrix of the possible interaction or the beneficial or harmful or neutral between species 1 and 2. The names of the interaction A, B, C, D are given respectively. Here C stands for the one species having beneficial for both, so it stands for mutualism. And number B, here one is benefited and other is harmed, so it may be due to the predation. And number A, here both are having the negative effect, so it might be due to the competition. And number D, where one is have a negative effect and other has no effect, so it will be the amensalism. So the answer will be number 3. Yes, question number 126. Which of the following is or are not valid explanations for the observed pattern of species richness? Here we have to find out that which statement is not will not favor the richness of a species. First statement is older communities are more species rich. It's obvious, because the community as it grows older and older, it will learn to adapt to its adverse environment, and also some genetic mutations also occur among the species which make them rich accordingly to their environment. So the statement is true. Here we have to find out the statement which is not valid. Second is larger areas support more species, obviously, because if the area will be larger, then the number of species will be more, because the sources for their survival will be definitely more to support their life. Next statement is natural enemies promote reduced species richness at local level. We can't say that the natural enemies will promote or reduce the species richness, because there are a certain food webs or certain patterns of survival at a particular area is there, which also consists of the prey as well as predators. So if it is a natural enemies, then it is the natural mechanisms for the richness of a species, or we can say for to adapt at the particular environment, which will also favors uh, its its distributions in a area. So the natural enemies will not promote the reduction in species richness, rather it will balance the richness of species in that particular area. So the statement is wrong here. Next statement is communities is in climatically similar habitats may themselves be similar in specific species richness. Obviously, every each species has its own criteria of survivorship and their own criteria to adapt an environment. If a particular species is getting the suppose a source of A environment, then if A environment is in this particular area also, then this species 1 will also be rich in species 2 also. So it will be similar in species richness. Next is greater productivity permits existence of more species. It's obvious, if we will cook more food, it means some guests are coming to our home and we can feed to more guests. Likewise, if there are more productivity in a particular area, then there are more species will able to exist in that particular area, or the productivity will increase the existence of more species. So only statement wrong is number C, so the answer will be only C. Next question is, yes, it says from the Shannon's diversity index, we have to calculate the Shannon diversity index. Here there we have two communities 1 and communities 2, and the A, B, C, D are the different species, and this value denotes the abundance of that particular species in community 1 and community 2. So here we have to calculate the Shannon index in the community 1 and community 2. So what is the formula to calculate it? Actually, diversity index H is equals to summation of, here we will see that I, or the number of variable, or the number of species, or the number of abundance, I may be equals to 1, or may it increase up to the level S. It is equals to the summation of Pi, Pi log in Pi, multiplication of Pi into log in Pi, and where Pi is the proportion of the population made up of species. What is the proportion of the population made up of species? And S is the total number of species in a sample. So total number of species in community C1 is 100, and in community C2 it will be what it will come? It will be also 100. Yes, 10 and 100. So species total number of population is same. Now if we have to calculate the Pi, then what we will do here, it is the proportion of the population. Then what is the proportion of A in total proportion 100? Then how we will calculate the proportion? We will calculate the proportion as 25 by 100. This is the proportion of species A in total among the total number of species. So it will be 0.25. Similarly, for the all, we will calculate the proportion of the populations of different species. Now, sorry, let me take another page. Here we have abundance of C1 and C2 species. For community C1, we have abundance of different species A, B, C and D, and all are similar 25, 25, 25 and 25. Now we'll have to calculate PI. How will we calculate the proportion of the population? I have told it I will divide the total number of population with that the number of particular species. Then it will come 0.25, 0.25, 0.25 and 0.25. Now what will the login of PI? Here let's see what is given here. Let me back to the question. The value of PI will be for 2.5 it is 1 2 3 it is minus 1.4. So let's put the value of login here, minus 1.4 minus 1.4 minus 1.4 and minus 1.4. Now when we will multiply here PI into login PI, we will get minus 0.35 minus 0.35 minus 0.35 and minus 0.35. And what is the formula for Shannon's diversity index? It will be negative. So we will get the sum of PI into log in PI. So it will be, when we will sum it, it will be sum of PI into log in PI, and it gives the negative value. So when we multiply with negative, we will get the positive value. So then sum of this is 1.4. Now back to the question. As per this, for C1 it is 1.4. Also let's calculate for C2. For C2 community, the abundance for A, B, C and D is, here it is given 80, 05, again 05 and 10. Its total number is 100. PI will be 0.8, 0.05, 0.05 and 0.1. Now let's again take the login value of 0.05, 0.1, 0.8. It will be, the login value will be minus 0.2 as per the question, minus 3.0, minus 3.0, and for 1 it is minus 2.3. Now when we will multiply with log PI into log PI will be, it will be minus 0.16, minus 0.15, minus 0.15, and to multiply with 0.1 into minus 0.2 to 2.3 will be 0.23, and the sum of this, so negative sum of this PI into login PI will be what? It will be 0.69. Very simple question was that, but only we needed to know the formula. So the answer will be number 1. Hope you understood it very clearly, and it's quite simple questions. You can very easily score 4 marks in such questions. Move to the next question. Again, it's numerical based on the biology, that is the Hardy-Weinberg equilibrium. In a population at Hardy and Weinberg equilibrium, the genotype frequencies are, for here the frequency for A1 is 0.59, for the heterozygous A1 A2 is 0.16, and for homozygous A2 is 0.25. Now here we have to calculate the frequencies of the two alleles at this locus. It means we have to calculate the value of P and Q. So what is the value of P? We know that for P, suppose we take the frequency P for A1 and Q for A2. Then I already have discussed in my video while I have explained the Hardy-Weinberg equilibrium. You can also refer that video, but this time I am directly coming to the solution of the question. I am not explaining much about this. If you want to tell, then please refer to that video in my channel Be Yourself. So the answer will the formula will be Total number of frequencies of A1, A2. Here it's the 2, it is in the homozygous state. So I will multiply it with 2 into 0.59 plus here A1 is only one time. So here it will be plus of 0.16 and if you will sum up all this we will get it 1. So here 2 into total number of population is 1. So I will take the, when we will calculate it, we will have the value of P as it will be equals to 0.67. Now to calculate the value of Q, what we do, we will minus it. We know that P plus Q is equals to 1. So the value of Q will be equals to 1 minus P. That will give 1 minus 0.67 will be equals to 0.33. So the answer will be number 3. Next question. Homing pigeons, when released at a place far away from their home, use Earth's magnetic field or the sun as navigational clue and choose the right direction to fly. To test the hypothesis, two experiments was conducted in experiment number 1. Here we have the test and control. One group of the pigeon test were equipped with the Helmholtz coils which disrupt magnetic field direction, while the second group, which is the control one, were not. It means the test pigeon will not able to track their home with the help of magnetic field. So for the test, the magnetic field, here it's the magnetic field and other is the sun navigation. So control don't have any magnetic hindrance, so it will able to track their home with the help of magnetic field as well as with the sun navigation, but the test will not able to track their home with the help of magnetic field but can track with the help of sun. Next experiment is in experiment number 2. In experiment number 2 we have test and control, and same situation for the magnetic field as well as for the sun. Okay, use the same control and test groups of pigeons, but they were released on a cloudy overcast day. It means both the test and control will not able to track their home with the help of sun because the environment is the weather is cloudy, so it will not able to track neither test nor the control one. So then expected result if the hypothesis is true. It means same, here we are using the same test and control. So in the test I have it was given that it was equipped with the Helmholtz coil, so the test will not even able to track their home with the help of magnetic field also, but control one can use the magnetic field to track their home. So what will be the possibility that in the group 1 or the experiment 1, both test as well as control will able to arrive their home, but in experiment number 2 only control will able to arrive their home. So let's see the option. In experiment 1 both control and test groups fly in the right direction, it's true, but in experiment 2 only group control groups does, it's true. So the answer will be number 1. Yes, next question is brothers A and B have the same father but different mother. If A and B will be from the same father and mother, then the relationship or the cost or the benefit will be, or we can say the relationship between A and B will be 50% and 50%. But as it's from the same father but different mother, so it will again reduce to half. So B will the relations between A and B will be again reduced to half. That is, it will be 25%, or we can say 0.25. So the R or the relationship between A and B will be 0.25 and have the same father but different mother. B wants A to help him, which involves both benefits and cost for A. If A incurs a cost of 30 Darwin fitness unit in that act, under what conditions should he help B following Hamilton's rule? So we know that to follow the Hamilton's rule, it should be R into B should be greater than C. So here cost is 30. So and B, and here we have to form between the relation between B and C. So B should be greater than C by R. C is given 30 and R is given 0.25, and when you will calculate this, you will get B should be greater than 120. So the answer will be number 3. Next question is from evolution. Following are the main types of defense employed by prey species against predators. The different types of defense mechanism and the prey species which adopt them. This question is quite simple. Only the thing is that you should know the terms related with that and the species which is related with that number types of defense. The first defense is chemical and aposematic colorations. This is shown by the field lizard, or sorry, this is shown by chemical and apometric coloration is shown by number. It's the Bombardian beetles, ladybird beetles, and many butterflies. What they do, they release certain chemicals which make them escape from the predators. So, E go with number 3. Cryptic colorations means the colorations according to their background. These types of adaptation is shown by grasshopper and seahorse. You have seen the grasshoppers mainly sit on the green leaves or green stems, which make it unable to distinguish, and also their leg and the antenna or the mouth structure is very much similar to that of the leaves. Their wings are very much similar to their wings. It looks like the stem or the tiny stem or sitting, or this is the part of the leaves. So its color is according to the environments, which is not cryptic colorations. So it will be shown by number 1, which is the grasshoppers and seahorse. Batesian mimicry is the one if a species adapt to not to fight against the one species, then it obviously adapt with the one which is similar to that. So such type of mimicry is adopted to it. For example, some poisonous snakes, non-poisonous snakes resembles to that of the poisonous snakes. It is known as Batesian mimicry. So it will go with that of Batesian mimicry. It's go with hoverfly and wasps. Actually, what happens? Some birds doesn't feed on the hoverflies, and wasp is not for them. So once they have adopted not to feed on the hoverflies, then obviously the wasps will be get benefit of this. So the answer of a Batson mimicry is number 2, and the intimidation display is sometimes, as in lizard and some porcupine fishes, if you will look the porcupine fishes, the tail has some eye spot. It's the tail. Uh, I'm drawn very badly. The fish. It also resembles the fish. It has fish. Uh, the tail also have eyes. So how it protect itself? What happens when anyone see its tail with the black spot? They predict that it's a eye or the fish is facing toward this side, so it will attack toward this side, and the fish get intimidated of the attack and able to escape from the predator. So such types of such type display which is not the eyes but it is displaying like the eyes is known as intimidation display. So the answer will be number 3. Next question is, yes, the it's from the evolutions. Following is the list of some important events in the history of life and name of the epochs of Cainozoic era. I'm directly telling you the answer because this is question based totally based on your memory. You have to mug up the chart of the evolution to solve such questions, and though it's take time to mug up, but if you will revise once in a week, then you will able to solve such questions, because if such questions you will attempt and it is in the 4 marks, then you will definitely score the positive. It will never go negative. So I'm directly answering the question. It's number 3. You yourself match it and mark up the all the points. Next question is, it again from the evolutionary tree. given below. The term A, B, C, D, E represents respectively. Here it goes from the lowest to the highest. The today without no any dot, we are the today's evolutions, and it is from the mammals. So we are the today's evolutions, and it will be definitely Homo sapiens sapiens. So and then it will again B C B D will be somewhat similar to E, and C will be far apart, but will have the same ancestor. So the D will be yes, it will be Denisovan, and C will be Neanderthal, and B will be Homo heidelbergensis, and A will be Homo erectus. So the answer will be number 1. Next question is, in the transgenic mice, the orientation location of localization site determines whether recombinase introduces a deletion, an inversion, or a chromosome translocation. If a researcher wants to put the localization site in such a manner that only inversion will take place, which one of the following constructs best justifies their intention? Gene X in the target gene. So, if gene X is in the target gene, suppose it has the somewhat like ATGCC sequence, then what we should get the resultant, we should get resultant as CCGTA, and this is known as inversion. And gene X is the target gene, and if we want to control it with the help of LOXP. So if the gene is outside the locks is not in between, then it is not possible the inversion in between. Again here, if the best describe, here we have to choose the construct which best describe the inversion. The best thing is that if it is inverting, then it should be in between these two genes, and these two options has somewhat confusion. One is that this, if suppose we take it as 3 prime and this is 5 prime end, then it will be 5 prime. Again, as per this, it is 5 prime and it is in the same direction. If we will invert it, what will happen? This will invert in this directions and it will go wrong. Or we can see the 5 direction will be pointed. This pointed side will be somewhat like this. It will go 5 prime and it will face the 3 prime. But in this diagram, if you will see, if it will undergo inversion, then it will orient themselves in the right direction, which is the 5 prime and 3 prime. So the answer will be number 3. Next question is, the following genes have been genetically engineered to develop the herbicide resistance in plant. We know that CKMet3 phosphate synthase genes are involved there in the for the herbicide resistance. So the answer is A is right. Another is Balaphos resistance using the bar gene. There is no use of bar gene. Sulfonyl urea resistance using the acetolactate synthase. No, it's yes, it's we will use the sulfonyl urea because it is a herbicide resistance. Okay, so the answer will be A and C, so it will be number 4. Next question is, yes, factor 9 is essential for blood clotting. Deficiency of factor 9 using would be corrected by delivering factor 9 gene using viral vectors. In an experiment, the gene for vector 9 was cloned appropriately into adeno-associated virus, and retrovirus. Retrovirus, it means we have used the 3 virus AV, AAV, and RB, and here we have successfully introduced the gene for factor 9 formations. But another statement says that retrovirus integrate the gene into the dividing cells. If it is integrating the genes in dividing cells, then it means all of the cells has been introduced with the factor 9. So it will express the vector 9 in more appropriate way. But adeno-associated virus integrates partially into the non-dividing cells. So after a certain time, the gene or the function of gene factor A9 will be abolished because it is not passing to the dividing cells or to its daughter cells. So it is partially integrated into the non-dividing cells. So it will last very less time. If it does not integrate the gene but transfects both dividing and non-dividing cells. So it is not integrating, but it is infecting both dividing and non-dividing cells. Now the point is that if it will be present in non-dividing cells, then as far as the adenovirus will be there, it will be expressed in both dividing as well as non-dividing cells. But once if adenovirus will not be inherited, it will not be carried to the daughter cells. So after a time, the expression will definitely decrease. So if we will see this, the answer will be A. A denotes as it is for the non-integrated, or it is partially. If we will see adeno-associated virus integrate partially into non-dividing cells, then once it has been introduced in the adenovirus and what it is in the non-dividing cells, it will first express high and then it will drastically decrease because it is not passing to the other cells. So A denotes the adeno associated virus, and B, it will last for longer time because it is integrated, as it is integrated the gene dividing cell. So it will last for the long time. It will be retrovirus. But as the AV virus is not integrated but it is present in both dividing and non-dividing cells, then after a certain time when it will not be inherited, it will be the production for factor 9 will be decreased. So the number C denotes the AV. So the answer will be number 3. Next question is, somatic embryogenesis is an important exercise in micropropagation and genetic engineering of plants. For following steps are considered as critical for achieving somatic embryogenesis. For the somatic embryogenesis, we will reduce the concentration of sucrose in medium to half. No, it's not there. Addition of hormone 2,4 dichloroacetone to induce somatic embryogenesis is true, and another is that we use maltose instead of sucrose because it is the more best source for the carbohydrate as compared to the sucrose. So the answer will be number B and D. Number 2. Question 138. For the aquaculture farming of Indian major carps, several techniques are used. Which one of the following techniques is not used for the purpose? Induced breeding is selected, selective breeding is selected because we have to select the hybrid ones for the better result. Inbreed, if we are using the inbreed, then it will decrease the quality of the hybrid, as well as it will reduce the offspring also. So the answer will be, which is not used in the technique, is the inbreeding, which is number 3. Next question is, the gene from the genomic library is screened by using the hybridization techniques. After hybridizing the probe using a stringent washing step is given, the following statement is given to explain the stringent washing steps. So what does stringent washing is means? I explained in while have explained the Western blotting, that if we these are the test samples and we have to find out the sample which whether it is there or not. What we do, we will design an antibody against it. So the antibody which is there which will bind to the test antigen, and there can be possibility it will not bind to the test antigen. So when we will wash out it, then what will happen? The unbound or the non-specific antibody will be washed out, and the only left here we will have the test or the antibody which is bound to the test solutions. And now from there we can study the further. For the west, it can be western blotting, northern blotting, or the southern blotting. So what will happen here? It is using the hybridization technique. So in this case also, while we will wash out it, then it will remove the unincorporated and the non-specified hybridized prop of the molecule, and it is usually carried out at the low temperature and at the high temperature and low salt, because if the temperature will be very low, then the hybridization will be very non-specific. But if the temperature will be high, then only the specific sequence will able to, or with the one which is complementary, very much complementary to the hybrid one, can only able to bind with the strand. So it will give the high specificity. So the stringent should be high it should, and the concentration of salt should be low, and temperature should be high. So answer will number C. And the salt present in the washing solution support hybrids to stay contact in shielding the interface of water molecules. Yes, the salt should favor the interaction between the hybrids, not it should remove out it. Next is salt reacts with DNA. No, it doesn't react with DNA. It just is provide the platform for its interactions. Next is that stability of hybrid is directly proportional to the temperature. No, stability even if the temperature is very high, then also it is not possible for the hybrid to form. And if it will be very low, then it will bind non-specifically. So it doesn't have any proportion with the temperature, but it works specifically at a specific temperature. So the answer will be A, C, and D, number 2. Next question is, a student while constructing knockout mice selected mouse embryonic stem cells and introduced an engineered DNA into the cell. However, none of the mice were transgenic. On checking the cell containing DNA construct, he found that he had made a mistake in constructing the DNA, since the cells were resistant to gan- ganxaclovirus but resistant sensitive to G418. Which of the following construct had he designed? The design, actually he does a mistake while designing the strand, because of which it enables to integrate into the DNA. So this is the site of homology, and if it has if any of the sequence will already be inside this in between the site of homology, then it will not be resistant to the ganocyclovirus. So the condition is that all the insert will be outside the construct. Because of this, the transgenic mouse haven't shown the positivity or the success of the experiment. So the answer will be number 3. Next question is, in the confirmatory test for HIV, one or more virus antigens were detected in the blood of the patient. Following are the steps in to be performed for the experiment, and the experiment will be again based on the antigen antibody interactions. What we do first, we will separate the viral antigens by the SGS base. Then we will transfer the viral antigen to the nitrocellulose membrane. After that, we will incubate it with the buffer containing the, let me mark it, 1, 2, incubate with the antibody specific for the viral antigens, and if the virus will be there, the antibodies specifically will bind with the virus antigen, and then we will detect the band or by the use of ELISA, enzymatic immunodeficiency immunosorbent assay, and will detect the secondary antibody whether it is present or not. Is number 3 and number 4. So the answer will be C, B, A, D, and the option will be number 3. In my upcoming videos, I will explain all the techniques very clearly. So at this time I'm skipping, at this time giving overview of this. But in my upcoming videos, I will explain it animatically, how this ELISA and how immunofluorescence, how precipitation takes place. So move to the next question. Yes, in most important property of any microscope is its resolutions, and can be calculated from the formula D is equal to 0.61 lambda by n sine alpha, where D is the minimum distance between two distinguishing objects and lambda is the wavelength of the incident light, alpha is the angular aperture, and n is the refractive index of the medium. Given below are several suggestions to improve the resolutions of microscope. We know that what we use the oil to increase the to improve the resolution of microscope, and also we decrease the wavelength of incident light so that the resolutions will increase. So what will be the answer? It will decrease the wavelength of incident light, as well as we will use the oil which has a high refractive index. So the answer will be number 1. Yes, next question is the mean and standard deviation of the serum cholesterol in the population of senior citizens are assumed to be 200 is the mean and 24 ml/dl per dl is the standard deviation respectively. In a random sample of total number of random sample or the n is 36 senior citizens, what value of cholesterol to the nearest whole number we have to find out the nearest whole number would lead to the rejection of the null hypothesis of at 95% confidence level. So for the rejection of the value of cholesterol level, we should have mu should be either greater than x bar or the mean minus error, or it should be less than mean plus error. So here first mean is given, here we have to find out the error, and to find out error the formula is error is z into standard deviation by under root total number of given sample. So for value, or we know that for value at 95% confidence or at 0.05 level, we know that z value of z is 1.69, and all the other values given as is given and is given. So the error will be z is 1.96 into standard deviation as is 24 by under root 36. When you will calculate it, we will get 7.8, and here it is given the nearest whole number, so that is expressed, it will be near to the approximately it will equals to the 8, and the here mean is 200. So mean minus error is here, it will be 100 minus 200 minus 8 will be 192, and plus 8 it will be 108. So the for the rejections of the for rejection of the null hypothesis, it should be greater, it should be less than, because if it will be greater than 198 then it will be accepted. So it for the rejection it should be less than 192 and it should be above the 208. So the answer will be number 4. Next question is, in response to a drug, changes in the protein level were examined in the cell line. A pulse chase experiment was performed using S35 label methionine in comparison to untreated sample. The following observations were made few minutes after stimulation. Protein X accumulates, and this was followed by reduction in the protein Y and Z. The correct interpretation of the observed would be, we can't predict it because the information is insufficient. It is not telling where it is binding or not. So the information is not sufficient to distinguish between these possibilities of the above statement. It can be through the feedback mechanisms, or it can act as a repressor for the transcriptional factor, or it can be act some toxins. So it is not clearly mentioned. So the answer will be number 4. Last question of part C is that the protein undergoes post-translational modifications in an experiment to identify the nature of modification. Following experimental results were obtained. One is the protein moved more slowly in the STA space, as selective focusing shows that there is no change in PI. Mass spectroscopic analysis showed that the modification was on serine. So first is that if protein is moving more slowly, then its molecular mass has been increased. And if it is not affecting the isoelectric point, then there is no addition or deletion of any amino acid from the protein. So its PI value is maintained. So it means some additional group is added to the protein because of which the molecular mass has increased, and this modification is occur during the serine residues, and we know that glycosylations occur in the serine residues, so the answer will be glycosylations. In detail I have explained in my channel. Be yourself, glycosylation topic is there. You can concern that topic too for your CSIR or another GET examinations. So all these were explanation in this part of second part of the CSIR JRF net part C. In first part, I have explained rest of the questions. You can also see that questions and answers if and in my upcoming videos, I will explain some more questions of the previous year questions from the CSIR, JRF net as well as from GET and DBT2. So don't forget to subscribe my channel. And one more thing is that I will also, as you have 1 month is left for your CSIR examinations 2017. So this time I will also explain the topics which is expected to come in this time. So don't forget to subscribe my channel by yourself and definitely share it so that more of you can benefit it, and click on the bell below the video so that you can get the notification as soon as I will upload my videos.